Hello and welcome to this revision video for Biology Paper 1. Topic 3 is called Infection and Response, and we'll start off by talking about pathogens. All these four examples of pathogens, and what a pathogen is any microorganism that gets inside your body and makes you feel ill. So for example, that could be a bacteria, could be a virus, it could be a protist, or it could be fungi. With a bacteria, these are small cells, and to make us feel ill, these bacteria produce toxins. A virus is not a cell, and it is not living. This is just a specially shaped protein. And what they do is they replicate inside your cells, and then they burst out of the cells and rupture them. That is what makes us feel ill. So for example, if we draw one of our body cells here, we're being the host, the virus is inside replicating, and then after a while they will cause that cell to then rupture and release the virus to other parts of the body, and that part causes us to feel very ill. Protists are single-celled eukaryotes. You can see that they've got organised parts inside them to make them a eukaryote. So single-celled eukaryotes. Some of these protists are often parasites that live or have part of their life inside of a host. For example, the malarial parasite that lives inside, the malarial protist is a parasite that lives inside mosquitoes. Fungi come in a variety of form. Some are single-celled. Some form long finger-like strands that come away, and those are called hyphae. You probably won't need to know that name in the exam, but some are made up of hyphae, which are these strands like things in the picture there, and some produce spores. The three main ways in which these pathogens are spread and that you would want to look to reduce, they are spread through water, they can be spread through the air, or they can be spread through direct contact. Pathogens then are responsible for communicable disease. So communicable diseases are ones that are spread by pathogens and that can be passed on from person to person. So one of the bacteria that you need to be aware of is Salmonella, because if you contract Salmonella, you can get food poisoning. So many of the chickens and the poultry market that is producing eggs, for example, they would all be vaccinated against Salmonella to try and prevent this, because this can cause food poisoning, which has the symptoms of diarrhea and vomiting. The other bacterial condition that you need to be aware of is gonorrhea. And this is a sexually transmitted disease, and the symptoms of this are that it causes pain when urinating and also a yellow or a green discharge. There's three viruses you need to be aware of. One of them is actually for plants, so let's start with that one first. For plants, it's the TMV virus, or tobacco mosaic virus, and what this does is cause a discoloured mosaic pattern on the leaves of plants, and this reduces the amount of photosynthesis that they can do. So for example, you'd expect this leaf to be all green and full of chlorophyll doing photosynthesis, but if it's contracted the tobacco mosaic virus, it'll end up having patches on it where they're discoloured in a kind of a mosaic-y pattern, and that will reduce the amount of photosynthesis that plant can do. Humans then, or animals, they can get the measles virus, and that is transferred by sneezes and cough droplets, and that has the symptoms of a rash and a really high fever, and you can get vaccinated against that one. HIV is another really deadly virus. It can either be contracted through as a sexually transmitted disease or by people sharing drugs needles. It targets the immune system and it makes that weaker, and then it can lead to the really serious condition called AIDS. So AIDS leads on from contracting the HIV virus, and it only can be treated by antiretroviral drugs. So it's a really one that's very difficult to treat, and if it leads to AIDS, can ultimately end in death. So an example of a condition that can be transferred by a protist is malaria, and the malarial protist, lives in mosquitoes. Part of its life cycle is within mosquitoes. So to try and reduce your chances of getting malaria, you need to think about reducing your contact with mosquitoes, like using mosquito nets, for example. And also, as a government, you'd want to target the areas where the mosquitoes will breed to try and reduce their numbers. This causes repeating cases of fever, and it causes the person suffering with the disease to become very ill indeed. You can get fungal infections in humans, for example, athlete's foot and things like that, but they want you to concentrate on one in relation to plants, and that is the rose black spot fungus. So just like the tobacco mosaic virus, with rose black spot, that targets the leaves. It causes them to go yellow and drop off the plant, and therefore the consequence of that is that the plant is performing less photosynthesis, because that takes place in the leaves, and that affects the growth of the plant. So there are a number of ways that you would be able to control the spread of pathogens that cause these communicable diseases. For example, in the case of salmonella, you're going to be wanting to be hygienic. For the case of gonorrhea, you're going to be wanting to practice safe sex and using barrier methods. In the case of measles, again, you're going to be wanting to hygienic, catch those cough droplets so that they're not spread around. With malaria, you're going to want to target the vector that's carrying the disease, in this case, the mosquitoes. And in some cases, you will actually be able to develop a vaccine, for example, for measles, to try and prevent yourself from ever having or developing that disease in the first place. So, unfortunately for us, there are thousands of pathogens that are trying to get into our body and attack us every day. But luckily, we have several defences against developing diseases. Our first barrier is our skin. It acts as a barrier, and it also secretes antimicrobial substances, so anti against microbial microbes, to try and protect us from any pathogens that are going to come into contact with our skin. If the pathogens venture towards our nose, within our nose we have mucus and hair to trap pathogens. If we were to breathe pathogens in, as we breathe them in, they will head towards our lungs through our trachea and our bronchi before going into our lungs. So within the trachea and bronchi here, there are cells which secrete mucus to trap the pathogens again, just like you have in your nose, but there are also ciliated cells to move the mucus and the trapped pathogens out of the body. And these will look something like this. So these will secrete the mucus, and then on the end they will have these cilia, which are these hair-like structures sticking out the end of the cell, and these will help waft the mucus up out of our body. If we swallow any pathogens through the food that we eat, they will go straight into our stomach, and within our stomach we have an acid to kill the bacteria called hydrochloric acid, and that will help kill any pathogens that enter our body that way. Despite all of these initial defences, some pathogens still get into our body and still get into our bloodstream. So within our bloodstream we have cells called white blood cells, and there's some different types of these. Some of them can engulf a pathogen. We say engulf because these are just cells. We can't say eat or anything like that. We mean engulf. So here we have the white blood cell, for example, moving towards our pathogen, and that will simply come and engulf it and then break it down inside it. And this process of engulfing the pathogen and breaking it down is called phagocytosis. The second thing that white blood cells can do is produce antitoxins. They're to counteract the toxins that are produced by the pathogens. And the final thing that they can do is produce antibodies. And these antibodies are very particular shaped molecules that will fit onto the pathogen. So this is the antibody, and this which is stuck to the pathogen, for example this could be bacteria, well these on the outside are called antigens. When the white blood cells release lots of antibodies, they will go and fix onto the antigen like so, and they will be an exact fit, and what that will then do is signal to these other white blood cells to come down and do phagocytosis and engulf that bacteria. An extra step that we can take to defend our bodies against disease is to get vaccinated. So we said before that if a pathogen got inside our body, that white blood cells can produce antibodies to match the antigen, and that will attract other white blood cells to destroy the pathogen. However, this process of making an absolute match for an antibody to an antigen takes quite a long time, and by then the pathogen might have replicated lots and caused us to be very ill. So instead what we can do is give our bodies a trial run, and what we'll do in a vaccination is we will inject dead or inactive pathogens into our body. So for example, when you've had the measles virus, inside that vaccine is dead or inactive measles virus ready to go into your body. But the pathogen inside will still have the antigens. So your body will start to work out the particular shape antibody that it needs to make to fix onto that antigen. But you are not at risk from actually getting a disease from that virus or that bacteria because they are dead or inactive. So it's harmless to you, but your body has time for your white blood cells to make the very particular antibody to fit on that antigen, so that if you were ever to get in contact with that bacteria or virus again, your white blood cells would straight away kick into action and be able to produce the antibodies and protect you from those pathogens dividing and making you feel ill. So examples that you might be aware of, MMR for example, that is a very famous vaccine because that contains 3 vaccines in one. It's a vaccine for the measles, mumps and rubella. So this is one way that we have controlled a lot of communicable diseases, and we can prevent a lot of epidemics by putting in vaccination programs where we start to see a particular pathogen spread amongst a community. If your initial body defences haven't worked, or if your immune system and your white blood cells haven't been able to defend yourself against disease, then you might have to rely on drugs. Painkillers are drugs that don't actually treat the disease, but they only relieve the symptoms of a disease. For example, if you've got fever or a headache. Antibiotics are particular drugs that treat bacteria only, not viruses. Viral drugs are very expensive to produce and make, so you can get antiviral drugs, but they are very expensive and they are to treat viruses. There has been major problems and maybe major talk in the press about the overuse of antibiotics, leading to something called antibiotic resistance. Antibiotics are used to treat bacteria. So say we had a bacterial cell in our body. If that wasn't treated, that would multiply and multiply and so on until you very soon had hundreds or maybe even thousands of bacterial cells in your body. If you were to take antibiotics, that would kill the bacterial cells. However, there are problems in that the bacteria are becoming resistant to the antibiotics. So this is what is meant by antibiotic resistance, when the bacteria become resistant to the antibiotic and it is no longer effective. And this all comes about through mutation. So as this bacteria multiplies, or maybe as this one multiplies, you may get a mutation which causes randomly these bacteria here to be resistant to antibiotics. And that would mean that random mutation would cause a resistant population that could not be treated by antibiotics. And the main problems with this are caused by the overuse of antibiotics. We're just using too many when we need to let our own body's defences try and fight the problem first of all. And secondly, we're not finishing the full course of antibiotics. So for example, if we had this number of bacteria in our body, obviously there would be many more in real life. If we took antibiotics and they were killing this many of the bacteria, if we didn't take our full course and we left one bacteria or a few bacteria, that would then risk being able to multiply and mutate its DNA as it was multiplying. So that would then risk becoming a resistant strain of bacteria, and you'd have to start all over again with a completely different antibiotic. Many drugs originate from plants. For example, aspirin, the drug, comes from the willow tree originally, and digitalis comes from the foxglove. There are a couple of examples that you need to be aware of. Other microorganisms have been developed into drugs as well. For example, Alexander Fleming found that mould had a property in it that killed bacteria. And from that he extracted penicillin, and that went on to develop the penicillin drugs, and that was used then as a drug to kill bacterial infections. So the pharmaceutical companies now have developed hundreds of thousands of different drugs that are prescribed by doctors, but they all have to go through a very rigorous process to develop the drug. So this involves preclinical testing, okay, so that's before human trials, and clinical, which is the stage where you're using human trials. So in the preclinical testing, first of all, the drugs are tested on human cells and tissues. The next trial of that is again before the human trial. They will be tested on animals, and obviously there's ethics, right and wrong, about testing on animals that you'll be able to look into and discuss. But they're tested for three things: TED, toxicity. So you're seeing whether they are harmful. Are they causing the cells to be damaged, or are they causing healthy tissue to be damaged? Efficacy. Do they do their job? Are they doing what you want them to do? And the final one, dose. What concentration do you need to use for it to be effective? Okay, so toxicity, efficacy, and dose. And then after all of that, if all those trials are successful, you're going to have human trials. The first stage of the trial is actually on healthy volunteers. So these people don't have the disease at all, but they're tested on these people just to check that it works within the human body system. So they're given a low dose and they are monitored and checked to make sure the drug is working around the body properly. And then it will be tested on patients with the disease. But to stop the placebo effect, which is where people psychologically feel like they're getting better if they're given a drug, the type of trial that's used is a double blind trial. And what this means, because it's double blind, it means neither the doctor nor the patient know who has been given the drug. So, for example, if you had 6 people that were taking part in this double blind trial, at random, some of the people would be given the drug. So, for example, if it was an injection, some of the people would be injected with the drug, and the other people would still be given an injection, but this time it would just have a sugar solution in it, for example, not containing the drug. So these people here will be given the sugar solution, which we call a placebo, which is a fake drug, so it's not going to do anything to them at all. And these people here will be given the drug, and neither the doctor nor the patient knows who has received the real drug, and they'll monitor them and see if they improve. And then the final stage is for the whole study to be peer reviewed, discussed with different doctors to avoid bias and to make sure that everyone is happy that the drug does what it is intended to do. Topic 4 is called Bioenergetics, but really it's a topic that looks at two major processes, which are photosynthesis and respiration. Starting off, we'll look at photosynthesis, which is a process that takes place inside cells. This is the word equation for photosynthesis. Carbon dioxide plus water makes glucose and oxygen. As a symbol, it would be CO2, add H2O, makes C6H12O6 plus O2, which we'd balance by putting a 6 there, a 6 there, and a 6 there. So you need to know both versions. This takes place in plants inside these structures here called chloroplasts. Within the chloroplast there is a green pigment that is called chlorophyll. And it is the chlorophyll that absorbs the sunlight needed for photosynthesis. So sometimes on top of these arrows you might see sunlight and chlorophyll to show that both of these things are needed for this process. But ultimately the goal of photosynthesis is to produce glucose that the plant then uses as its food. This reaction is an endothermic reaction linking to the chemistry topic because it takes in energy. So the glucose that is made in photosynthesis can be used by the plant in a number of ways. One way is through respiration, which is simply the reverse of the photosynthesis equation. So respiration is glucose and oxygen making carbon dioxide and water and releasing some energy, which we put in brackets as well. So straight away the glucose that it made in photosynthesis, it combined with oxygen in the mitochondria in the process of respiration for growth of the plants. The second thing that it can do is use the glucose molecules to build larger molecules such as cellulose. And cellulose is a molecule that strengthens the cell walls of plants. Another thing it can make with glucose is amino acids that can go on to make proteins. But to make these amino acids, it also needs nitrates, which it takes up through the soil. Okay, so as well as the glucose and the nitrates, that's how we make our amino acids, which are the building blocks for proteins. It can also store the glucose in a number of ways. It can store it as lipids, so which are fats and oils, which are really our seeds of our plants. For example, if we had some seeds forming on the plant here, these seeds would store lipids, and that's what we can turn the glucose into. Alternatively, the glucose can be stored as starch around the plant, and it will do that because starch is insoluble. It doesn't dissolve easily, so it's easier to store within cells. For example, it could store it as a starchy vegetable underground. There are several factors that can affect the rate of photosynthesis that we call limiting factors. Those three limiting factors are light, carbon dioxide levels, and temperature. Those are the main ones, but also the green pigment, chlorophyll, is also a limiting factor that can affect how much photosynthesis takes place in the plant. Chlorophyll becomes a limiting factor when the plant has a disease, because disease like tobacco mosaic virus or rose black spot fungus, that disease affects the amount of chlorophyll in the plant, and chlorophyll absorbs the sunlight for photosynthesis to take place. So if a plant is diseased, then chlorophyll will come in as a limiting factor. But the main ones are light, carbon dioxide, and temperature. So light and carbon dioxide have a very similar shape graph, whereby as light increases, the rate of photosynthesis increases up until a particular point, and then the rate will not increase further and it will level out. The same with carbon dioxide. As the rate of photosynthesis, the amount of carbon dioxide increases, so does the rate of photosynthesis until it levels out. This initial point of the graph here, while it's still increasing, this is when, in this case, light is a limiting factor. Or in this case, carbon dioxide, this is the point where it is a limiting factor. That means if you were to increase or decrease the light levels at this point, you would change the rate of photosynthesis. But after that, it doesn't matter. If you were to invest more in more lighting, well, that wouldn't increase the rate any further. So at this point, we would say that light is not a limiting factor because it won't change the amount of photosynthesis that takes place. So this is not a limiting factor at this point here, and the same for carbon dioxide here. Temperature has a different shape curve, and that's to do with enzymes that control the rate of photosynthesis. So this here would show the optimum or the best temperature for photosynthesis to take place at, and then after this the enzymes denature and the active site changes shape and they will no longer work. So up until this point here, temperature is a limiting factor, and then after the optimum temperature the enzymes denature and no longer work. So this would be important in the winter. Temperature is the obvious limiting factor in the winter, and at night time light is a limiting factor because without the light you don't have photosynthesis. This extra slide is for higher tier only because you need to know how these factors can interact with each other. For example, if we had a graph that showed light intensity against rate of photosynthesis, before we just had one of these lines on the graph. So before we just had this one, for example. But you may well see in the exam graphs with two different lines on. And this could have a different key to show you that this one perhaps was at 15 degrees, for example, and this one was at 25 degrees C. And what this will show is that in this case, temperature was the limiting factor, because if you increased the temperature to 25, you increased the rate of photosynthesis. So temperature was a limiting factor affecting the rate of photosynthesis. Here we had one at 15 degrees that plateaued out, but if we increase the temperature, then the rate of photosynthesis also increased. This is not a difficult concept, but for higher tier only, you also need to know a little bit of detail about how greenhouses are managed. So greenhouses are glass buildings that are used to grow lots of plants, and the main trade-off that you have when running a greenhouse is the cost of running the greenhouse versus the crop yield that you will get inside the greenhouse, whether that is the flowers that you're growing or the vegetables you're growing or whatever it is. So you want to maximise the rate of photosynthesis that's taking place within the plants inside the greenhouse, but at the same time not be wasting your money when you're doing so. So this will link into limiting factors and the things that will affect the rate of photosynthesis. So obviously the outside is glass to try and let lots of light in, but you also might want to put artificial lights inside the greenhouse to increase the rate of photosynthesis. Obviously they're going to come at a cost as well. You can also help to heat your greenhouse, especially important in the winter, if you were to put a special heater inside called a paraffin heater. And this does two things. It will help maintain the optimum temperature for you, which is important, but the paraffin heaters also release CO2, so they'll help optimise the CO2 levels inside the greenhouse for photosynthesis. You can also have windows like these at the top, this top layer here, that can automatically open and close to adjust the temperature to make sure that is optimal. And of course the fact that it is all in one enclosed building will also reduce the number of pests inside, and obviously that is a added benefit for you when you're trying to grow plants inside. So think of perhaps an evaluate question associated with this, looking at the trade-off between the cost of these things and also the effect that they will have on photosynthesis and crop yield. One of the required practicals for this paper is looking into how we can measure the rate of photosynthesis. So we can do that by using an aquatic plant, because we can see gases that are released from these plants. So this is a pondweed, and when photosynthesis is taking place, the pondweed will be releasing oxygen and will be releasing bubbles from the leaves. And we can count these bubbles, and we can use this as a measure of the rate of photosynthesis, so how quickly photosynthesis is taking place. So that would be our dependent variable that we are measuring, and we can change a factor. For example, we could decide as our independent variable to have a look at light intensity. And if we wanted to do that, we could set a lamp at a particular distance from the pondweed, count the number of bubbles that are given off in a certain time frame, and then move the lamp a little bit closer, say to 8 centimetres away. Again, count the number of bubbles are given off, increase the light intensity by bringing the lamp closer, and so on and so forth. As an alternative, perhaps you didn't want to measure light intensity and you wanted to keep the lamp in the same place, you could have investigated a different factor. For example, how does the colour of the light affect the rate of photosynthesis? In this case, you'd keep the distance the same and put different colour filters over the lamp, or change the light bulbs for different colours. So for example, we could put a red filter over there and count the number of bubbles, and then we can change that to a blue filter, count the number of bubbles produced by the pondweed, and so on and so forth. A quick extra bit for higher tier, you need to know about the inverse square law in relation to light intensity and the distance between the lamp and the pondweed. So if we were to think about this, the basic relationship is that the further away the lamp is from the pondweed, the lower the light intensity reaching that pondweed. Whereas if you put the lamp up here, really close, you're going to have a high light intensity. But actually that has a very special relationship called an inverse square relationship, whereby the light intensity is proportional to 1 over the distance squared. So you need to be aware of that relationship, and you also might need to be aware of the fact that light intensity is measured in a unit called lux. Okay, moving away from plants and back onto animals and humans, we're going to look at metabolism. So metabolism is the sum of all the chemical reactions that take place in your body, and you may hear communications of some people saying that they have a high or a particularly low metabolism. So they're talking really about the rate at which these chemical reactions are happening in the body. Now, some of these chemical reactions might be reactions to build larger molecules. Some of these chemical reactions might be reactions to break down large molecules. The first type of respiration that we're going to talk about is something called aerobic respiration. This is the equation for aerobic respiration. It says that glucose reacts with oxygen to produce carbon dioxide plus water, and you'll also see plus energy in brackets, because energy is not a molecule, but it is something that is released in this reaction. If we put the chemical symbols in here, we've got C6H12O6 plus 6 lots of oxygen produce 6 CO2 plus 6 H2O. So what's happening in respiration is that it's the process that transfers energy from glucose, and it happens in every cell in the body. So the whole point of this is we break down the glucose that we consume in our food, and we are wanting to release the energy, because the energy can be used for a number of things. It can be used to build large molecules. It can be used to maintain our body temperature, our internal body temperature, using homeostasis to regulate our internal environment. And it can also be used importantly in muscle contraction for movement. So we need that energy, and we get that by breaking down glucose. And this is where it takes place in the cell, in these subcellular parts called the mitochondria. So aerobic respiration is the one that involves oxygen. Okay, so if you see oxygen, think to yourself aerobic respiration. And this is the one that is very efficient, meaning that lots of energy is released through aerobic respiration. This process happens in both plants and animals. Most people associate it with animals because we do respiration, but remember that this also happens in plants because plants need energy to build larger molecules and to grow as well. The other type of respiration is called anaerobic respiration, and in anaerobic respiration this is without reacting the glucose with oxygen this time. We only use anaerobic respiration in emergencies because anaerobic respiration is not as efficient. It releases less energy than aerobic respiration does. So we only use it when we don't have enough oxygen in our system. So here we've got the equation for anaerobic respiration in animals. What happens is the glucose molecules are broken down straight into something called lactic acid, and a little bit of energy is released. Now the trouble with this builds up inside muscles, and this can cause cramps. So we don't want to use this very often. We only use it in emergencies when we don't have enough oxygen in our system. Plants and yeast have a different form of anaerobic respiration. This is whereby they take glucose and they produce ethanol and carbon dioxide. And anaerobic respiration in yeast in particular is called fermentation. And the fermentation process whereby yeast takes glucose and produces ethanol and carbon dioxide is used in two major industries. It's used in the bread industry and it's used in the alcohol industry. Ethanol is the main alcohol that's used in alcoholic drinks. So the fermentation process produces ethanol, which is extracted and used to make alcoholic drinks. And in the bread making industry, it's actually the carbon dioxide that they're interested in, because the carbon dioxide helps to make the bread rise, because the bubbles of gas are forming in the bread and it helps to make the bread rise. This extra section on oxygen debt is for higher tier only, and this relates to the fact that if you have to do anaerobic respiration, you'll take glucose and you'll turn that into lactic acid and energy will also be released. Oxygen debt describes the amount of oxygen needed to break down that lactic acid and remove it from your cells. So I'll write the definition here. The amount of extra oxygen needed to break down the lactic acid and remove it from your cells, because you don't want it to be causing cramps. So debt and oxygen, it's the oxygen that your body needs to break down the extra lactic acid. And when it does break that down, that lactic acid, it will turn it back into the main products of respiration, which are carbon dioxide and water, just like you'd normally get from aerobic respiration. So to do that, at the end of exercise, you will be breathing heavily to try and get more oxygen into the blood. Both your pulse rate and your breathing rate will be higher after the race, to try and not only get that oxygen into your blood, but then your pulse rate high means it can be pumped around your body to your muscles where you will need it. And as an extra fail-safe, you've also got your liver that can be involved to help you as well, because if needed, the liver can convert lactic acid back into glucose and store it. So the blood can take the lactic acid back to the liver, and there it can convert it into glucose and store it. Respiration then links into exercise. So when you exercise, you will need to do a higher rate of respiration, because during exercise the muscles that you are using need to be able to contract more, and when they contract more they need energy through respiration. So muscle contraction uses the energy released by the mitochondria, so if you need more energy, that means you need to respire more inside your cells. In order for respiration, if we remember the equation, you need your glucose and your oxygen to make carbon dioxide and water, and obviously the energy released as well that we put in brackets. But we also need to have more oxygen. So when we're exercising, our breathing rate will increase. The volume of breath that we take in, our breathing volume will increase. And our heart rate will increase as well. So we'll try and get oxygen into our body and move it around our body, and we will also need a higher rate of respiration to release energy to allow the muscles to contract.